Okay. So we left off, we're talking about communication, right, in the endocrine system. And we talked a little bit about paracrine, exocrine, and that sort of stuff. And so that's kind of a little on the new side, right? So those different types of communication patterns. And then, of course, there's synaptic communication. You guys know a thing or two about this one, right? This is what we talked about with the neuromuscular junction in AMP1. And so this is typically any kind of a situation where you have to where you have that synapse setup, right? Where you have a presynaptic cell, and then that has to communicate to a postsynaptic cell, and there's like a gap there. So anytime there's like a gap that has to be jumped by these chemical messengers, right? These neurotransmitters, and that's a synaptic version of communication. So it's kind of like its own special setup that is probably in spirit similar to paracrine in nature. It's just kind of a specialized type of paracrine, right? Where it's kind of cell-to-cell communication through these chemical signals. And so for you visual types, this is your visual. So you can see here, you've got direct communication where you're communicating one cell to another directly through gap junctions. So this is for coordination of response. That's basically what you have here. And then of course, paracrine, where one cell will secrete like a hormonal signal, some sort of a chemical signal, and then it'll basically go and communicate to a whole host of different types of cells. Generally speaking, those cells are somewhere in the neighborhood, right? You're not talking about far-flung cells like halfway across the body. These are usually cells that are within a fairly close proximity to the original secreting cell. So this is like local sort of a situation. Now, a modification of the same basic idea, like say, for instance, if you need to go from a local to a distant kind of communication, then you move into endocrine, right? So endocrine is different than paracrine in the sense that instead of secreting your signals to adjacent cells, you're basically dumping them into the bloodstream for broadcast across the body. So in this particular case, your targets are far away, right? They're like a long, long ways away in the body. And this allows you to be able to kind of distribute these signals to those particular cells. And then, of course, you got your synapse, which we know quite well, right? Because the synapse structure took up 2 chapters in AMP1, right? So muscle physiology and neurophysiology. Okay, let's take a look at the hormones themselves, right? These chemical messengers themselves. So we have different types of hormones. Not all hormones are the same. Some are proteins, some are not. We start off with the peptide hormones because oftentimes those are the ones we usually think of first. They come in all shapes and sizes, right? So basically when you have these, and there's a benefit to being a protein type of hormone, because when you're a protein hormone or a peptide hormone, you have the ability to sort of structure yourself in a way that kind of has you in an inactive state, right? Which is really, really helpful when you need to make the hormone, but you don't need it to be ready to work right as soon as you make it, right? So you want to distribute it first and kind of get it all situated, and then you want to give it the get the go button for it, right? So in order to do that, you kind of have to make it in an inactive state so that you can position it and distribute it, and then you activate it, and then we're off to the races. And peptide hormones are particularly useful for that kind of strategy. You use a lot of that actually in the body. And so oftentimes we'll see these inactive types or versions of these hormones. We oftentimes will give them a particular prefix name, prohormone, right? So a prohormone is basically an inactive version of that particular hormone. Enzymes have a similar type of a strategy where you make them in an inactive state and then you activate them later. Only in that case, we have a different name for them called a zymogen. which is like an enzyme that's in inactive version, so you haven't turned it on yet. Those are particularly common, especially when we get to the digestive system, because a lot of our digestive enzymes obviously are hydrolytic, and so you don't just want those guys on all the time because they're just going to rip through your tissues. So you kind of make them in an off state, in a zymogenic state where they're off, and then you turn them on later at the right time in the right place. Right? So that's kind of so that's a common theme. The reason why I mentioned that is because this idea of making something in an inactive state so that you can distribute it and sort of strategically place it is a very, very common and a very important technique that you use in physiology for regulation purposes. Okay, and you see it across the board, not just with hormones. Okay, so what kind of proteins are we talking about here? Well, some of the peptide hormones are glycoproteins. And so these tend to be fairly on the big side. And we're talking about big is defined as something bigger than 200 amino acids. So that's kind of our cutoff point. A couple of examples of this would be some common ones that we see, like thyroid stimulating hormone, which we'll talk about here in a few slides. Luteinizing hormone and follicle stimulating hormone, both of which are involved in the female reproduction and also male reproduction, by the way. So just because LH and FSH have feminine associated terms to them, obviously males don't have follicles. They actually have function in males that are different, but that are also still involved. And so those are important, large glycoprotein types of complexes. So remember, a glycoprotein is basically a big protein with some sort of a carbohydrate sort of ornament or a branch on it. And that carbohydrate branch can have a lot of different looks to it, right? And sort of the look that you have is going to be dictating your specific function. And so you may have recalled from general biology, if you took it, the glycoproteins are the way that cells sort themselves out. It's the way they identify each other. Like it's the way two liver cells see each other as two liver cells, and they know they're on team liver. Right. So sports teams, they wear the same colored jerseys. With cells, they have the same type of glycoprotein that tells them, hey, I'm a liver cell. Let's form team liver and do what liver stuff does. Right. So that's kind of the way that works. So glycoproteins are a really important group of proteins in general. But what if you're smaller than 200 amino acids, right? So what if you're not quite that big? Well, we do have some hormones that are a little on the smaller side, not super tiny, but a little smaller. And so some of these will be super, super tiny. Like for instance, the short chain polypeptides, that's one type of small peptide. And we're talking really little. We're talking like little chunks. Of peptide. And by that we mean you're talking about like maybe less than a dozen amino acids. That's how big you are, right? So ADH is an example of this one. Oxytocin is an example of this one. You may remember that from the previous couple of previous slides ago when we said that the hypothalamus makes ADH and oxytocin. These are really, really short peptide chains, but they're really important. And the reason why I like to point that out is because a lot of times in biology, especially in physiology, we kind of get this sort of Texas mentality, right? Like go big or go home, right? Like all the good stuff is big. And so if it's important, you got to have a lot of it. And physiology is actually just the opposite. A lot of times the most important things are very minuscule. It's like a spice. It's like a really strong spice. It's like that's not the main thing. It's just like an accent that really gives the dish its character. Same thing is true here. Here's a really, really tiny peptide chain. If you didn't know any better, you would think that this is just protein garbage in the cell, like some degraded trash from a protein that got broken down somewhere, and you just haven't processed it yet. But in reality, it has titanic consequences. We're going to talk a lot about ADH pretty much throughout the and then, of course, you have some things that are somewhere in between, right? So in this particular case, you've got some small proteins. They're not quite as big as the big ones, right? So the 200, but they're certainly not little small chunks. They're somewhere between, a dozen or so and 200. And this accommodates a lot of them. So for instance, growth hormones tend to be in that range. They're kind of up there. closer to the 200 range. So we're not talking about, enormous differences here. Prolactin is another one. And then, of course, that's we're talking about just the main central hormones here. We're talking about the hypothalamus and the pituitary. That's largely what we're focused on here, and most of our examples here. But we know that there's going to be more hormones that are associated with other organs in the body that aren't the hypothalamus and the pituitary, right? So we know, for instance, that there's some of these organs that have primary designations in other systems, but they have an endocrine function to them, right? We saw that on our previous list where we had these secondary organs, and a lot of these will also secrete different types of protein hormones. Like, for instance, you'll get a lot of protein hormones in your digestive tract. You'll get some coming from your heart and your thymus. Remember, we talked about the thymosins. And so there's a lot of different types of hormones coming off of a lot of different types of organs. But the heart and soul of the endocrine system is largely hypothalamus, pituitary, and the immediate target glands, okay, who basically don't have anything else to do with their life than just be a gland, right? That's kind of what we think of as our core endocrine. And then beyond that, and the next ring out, or like more the peripheral version of the endocrine system would be some of these secondary organs. So what if you're not a protein? Is that possible? Sure. Our lipids. How do we know that? Well, because some of our most famous hormones are actually steroids, right? So steroid hormones, and we have a lot of those, for instance, estrogen. And testosterone. Those are two pretty famous hormones, right? Matter of fact, when somebody says, "Oh yeah, his hormones are raging," what are they usually talking about? Testosterone, right? And so this is what we usually think of as a hormone, but they're lipids, right? So what does that mean? That means they're going to be able to move around differently in the body. And so one of the big themes in general biology was how these molecules move into and out of the cell, right? Cell transport. When you're a protein, you're kind of on the big side. You're kind of limited in terms of how you can move around the body because you're sort of big and cumbersome. You can't just go waltzing right into a cell, okay? Because you're a little on the big side. So you need some help there. However, with lipids, because your membrane is made out of lipid, you can freely just pass through that membrane as you wish. And so that's a different mechanism. And so there's a lot of different types of steroid hormones. Typically, all of them are based on cholesterol. So cholesterol is actually the parent molecule that will give rise to estrogen and testosterone, which is one of the reasons why you have to have cholesterol in your diet. There's no such thing as having a cholesterol-free diet because you'd be dead if that was true. So you have to have some cholesterol because that's where we get our steroids from. Now we get these off. Obviously, we're going to get a lot of these androgens. So andro for man, that's basically I don't know what that is, Greek or Latin, for maleness or masculinity, basically what that is. So this is going to be obviously associated with the reproductive organs. You're going to be producing this from the testes in male, estrogens, which is the other steroid hormone, and progestins, which are going to be coming from the ovaries of the female. So you can see that the reproductive hormones are steroid hormones primarily. However, oftentimes we stop there, don't we? We think, okay, we're going to talk about hormones, so we're going to talk about the reproductive hormones. We're going to talk about testosterone and estrogen, and that's oftentimes where we stop the discussion. But in reality, those are probably the least important of all the steroid hormones. Because you can do whatever you want with estrogen and testosterone. And you can have some pretty powerful fluctuations in those. And you can live just fine. Okay. I mean, this is actually part, if you think about it, this is actually part of gender transition. Right, so we can take somebody who is a chromosomal male with testosterone, and we can administer high levels of estrogen to that individual, and we can feminize that person's body. And we know we can do that. We've done that. That's part of that whole transition protocol. It's not the first thing you do, but it's somewhere in the middle. So we know that, by the way, when you do that, you're living just fine, right? So it's not like a life and death sort of a thing. It's not like you have to die first or whatever. So it's not a life and death thing. And that's what I mean by these aren't even the most important, because you can have dramatic fluctuations in these spikes and troughs in these and still do just fine, survival wise. By far, the most important ones are the ones that nobody really talks about, right? So you see a lot of the steroid hormones coming off the adrenal glands. We're going to talk about that because we're going to unpack the adrenal glands a little bit more closely, along with the thyroid. So these are the corticosteroids. So basically what these guys will do is, along with some of the other components of the adrenal cortex secretion, is these guys are going to be responsible for your basal metabolism. So they have a lot of sort of housekeeping activities that are necessary for your body. They're not sexy, they're not exciting, they don't have the wow factor to them, but it's like something you have to do. It's like back in the old days, it's sort of like, this is sort of like the balancing your checkbook. Okay, is that a lot of fun to do? Not really, right? But somebody's got to do it. Now we just let the banks do it. But if we even have checks anymore, most people are like, wait a minute, I saw one of those in a museum once. That's kind of where it's going. But somebody has to do that accounting bit, right? And it's not fun. And there's no wow to that, but it has to be done because you have to know how much money you have, right? It's by far much, much, much more fun to spend the money than it is to actually do the accounting to see how much money you've got in there. Okay, and these are sort of those accounting steroids. And of course, the kidneys will make another steroid called calcitriol, which is critical. Some of you guys may remember that as the terminal point for that vitamin D pathway that you learned about is one of the functions of the skin. And we'll learn about calcitriol after calcitriol. So basically then what you have is all these very important steroid hormones along with the peptide hormones, all working in concert and all basically communicating with their target cells. Now, here's the problem. If you're a protein, you can circulate through the blood, which is mostly plasma. We're going to learn that in chapter 19 here. That's mostly water. But lipids are what? How do lipids feel about water? Yeah, they don't mix, do they? Right? They're non-polar, they're hydrophobic. So what would a lipid in your blood want to do? Just add some oil to water, right? What does the oil do? It stratifies, right? It immediately forms a little oil layer. It's like the oil is literally trying to get away from the water. Well, think about this for a second. If you've got lipid steroids in your bloodstream, because we're talking about endocrine now, right? Not paracrine and that, we're talking about endocrine. Isn't that going to be a problem if your blood is mostly made out of water? How do you manage to get lipids, which are necessary, to move through your water-based blood system without turning into an oil slick, which would be completely functionless, right? And this is the theme that we're going to also unpack in chapter 19. But because of that, you need special proteins, transport proteins, whose job it is to grab onto these steroids and transport them throughout your bloodstream. As a matter of fact, this kind of lipid transporting group of plasma proteins that are in your plasma represents a dominant percentage of the proteins that are in your blood plasma by far. So because they're able to circulate, they have a bit more of more staying power, right? So proteins tend to be a little more, they burn off easily. They need to make more to keep the effect going, where steroids tend to stick around a little bit longer, so you can have a more enduring, long-term type of an effect. All important things to know, by the way, when you're trying to coordinate and create a particular type of effect, right? So it's like, what kind of effect do you want? Do you want a quick flash, right? Or do you want a long, slower, enduring type of a thing, right? So there's going to be different ways you set that up. So let's take a look at some of the reflexes that are controlling some of the endocrine system. So an endocrine reflex is similar. It's kind of like the cousin to neural reflexes. You guys remember neural reflexes from AMP1? A little bit, sort of. It usually goes very quickly at the end of the semester because we're running out of time usually in AMP1. But remember, the normal transmission of a nervous impulse is sensory first, right? So you sense something, and that goes through the afferent system, the afferent side of your peripheral nervous system, which basically takes that sensory information up to the brain. The brain makes some sort of assessment about that, right? You make some sort of a decision about that. And then it sends the response out of the efferent system, exiting, and then that goes down to your muscles, and then you basically do something like take your hand away. I don't like the way this feels, right? That's your normal transmission. It's driven by your brain. Input, interpretation, output. Now, a reflex is a short circuit of that system, where basically instead of waiting for the brain to make a decision about that, you immediately go to the spinal cord, through an interneuron, and then right back out of the hand. This is helpful, for instance, if I were to put my hand on an exceptionally hot surface, like a blazing red hot stove. I don't have to tell my brain or wait for my brain to make a decision that I need to move my hand. It just goes immediately right to the spinal cord and right back out to my muscles, and I pull it away before my brain even knows what's going on. Typically, reflexes are autonomic. They're not voluntary. And they're designed, generally speaking, to save you, right? So in this particular case, your reflex is designed to save you, because if you have to think about whether or not you want to move your hand is already half melted and for the most part functionless. Bad strategy, right? So you want to move this along a little faster. That's what a reflex does. In the endocrine system, reflexes basically, they control and regulate physiological autonomic activities. That's typically what a reflex is. It's like something where it's like, you know what, I need this to happen this way, but I don't want to have to think about it all day long. What you do then is you create an endocrine reflex to take care of that. Okay, we're going to take a look at this in a second. Now, one way that you set up a neural reflex is by setting up a regulatory strategy called a feedback loop. There's both negative and positive feedback loop. Generally speaking, whenever you get some sort of a sensory input, that's going to basically trigger this reflex, which will generally produce some sort of a hormone. And then that will basically act or operate immediately on whatever that reflex is designed to do. Okay, that's a lot of words for not a whole lot of meaning. Well, let's take a look at what we're talking about here. And I want to take a look at homeostasis, right? The entire idea of homeostasis is driven by endocrine reflexes. Now, that's important to draw this distinction, because remember, we started off the semester by saying you have two integration systems, right? Neural integration, and then you have endocrine integration. And so oftentimes you may look at that, you may think, well, wait a minute, these are overlapping, right? So these are redundant and overlapping. So why would you have two, right? Well, it's because they're not really doing the same thing. Think about this for a second. A nervous impulse or a neural reflex is designed very specifically for a very specific target. Predominantly, those targets are muscular in nature, maybe glandular, but predominantly muscular in nature. And they're very specific, right? So they have a very specific action to them. And not only that, but they're very temporal. So that nervous impulse, that stimulus, doesn't last forever, right? It's a stimulus, it's gone, you act and it's over, and you reset the system and you're ready for another stimulus. So what you're doing is creating movement. Now, the way endocrine reflexes are different is because what they're designed to do is they're designed to take a particular metric in your body, a physiological metric in your body, and essentially regulate it. It's acting like a thermostat. These reflexes act like a thermostat. So let's take a look at this. Let's use blood pressure, something we're going to be talking about for a good chunk of the semester. Because blood pressure is not simple. It's simple to measure, but it's actually quite multivariate. And we'll take a look at all those variables throughout the semester. But imagine your blood pressure, right? So your blood pressure throughout the day is under homeostatic control. So what that means is that during certain parts of the day, your blood pressure will rise and it'll peak. And then there is a reflex at the peak that will then deflect that curve back down. And then it'll trough. And then there are trough reflexes that will inflect that curve and send it back up. And then the peak reflexes will kick in again and push it back down. And then the trough will kick in and push it back up. And then this is you all day long. Back and forth, ebb and flow, just like a tidal in comes a tide, out goes a tide, same thing. So who's at the peaks? These endocrine reflexes, hormones, molecules, mechanisms that are designed to turn these homeostatic peaks down. Who's at the troughs? A counterbalancing reflex. We saw a little bit about this already, right? AMP1. Remember when we talked about calcium homeostasis? We said that calcium homeostasis is governed by two antagonistic forces. On one side, you have calcitonin, right? Which is kind of moving things back and forth. On the other side, you have parathyroid hormone, right? So you have those two going back and forth. And so that's always true. So this is like what I call the homeostatic dyad. You have flexors and you have the trough reflexes. And then throughout the day, your blood pressure stays within this relative norm. Okay. And so that's what an endocrine reflex is designed to do. It's designed to maintain homeostasis. So as a matter of normal activity, your endocrine reflexes are constantly going all the time. So let's take a look at the different triggers for an endocrine reflex. A couple of different types of stimuli. The first one is the humoral. Now, what the humoral stimulus kind of is basically a situation when you are looking at the extracellular fluid. So here's your cell. The extracellular fluid is outside the cell. So the makeup of that fluid outside of the cell, when you have changes there, that could cause an endocrine reflex, some sort of a change in the situation. Hormonal stimulus. So this is basically hormone signaling. By the way, exactly like we just saw in the previous slides, either communicating in an endocrine fashion or in a paracrine fashion or something of that nature. So that would be the hormonal stimulus. And then the last one is neural stimulation, right? So this would be the synaptic. Communication. This would be paracrine and endocrine. And humoral, if you're wondering what humoral would look like, this oftentimes could also be direct. Because in that case, you're not looking at extracellular, but intercellular fluid, which is changing, and that's causing a change, or a stimulus, right? So that could be both. Those could be also direct. So those are the different types of signals that would happen. Now, in the terms of the nature of the reflexes, like how could you structure these reflexes? Well, you could keep it simple, right? Just one hormone, one response. That's an easy one to do. So when you need simplicity, doing a simple reflex is a good way to go. And there's a lot of, we'll see this in a lot of them, right? The heart, for instance, pancreas, parathyroid, digestive tract has a lot of these types of simple endocrine reflexes to them. But what if you need more functionality than that, right? What if you're an artist and you need more than just a couple of colors? You need like the entire color wheel, right? So then you're going to need more complexity to your endocrine reflex. In this case, there's a couple of ways you can do that. You can either add more steps or you can add more hormones. Either one of those will increase your complexity. Now, why are we mentioning this? The reason why we're mentioning this is because what we want to do is we want to basically lay the groundwork for our regulatory strategy, right? Because when you're talking about all these cells in your body, all your organ cells and things of this nature, I mean, your body is doing a lot of stuff just right now. It doesn't seem like it might be doing a lot of stuff, but it's doing a lot of stuff. And all that stuff has to be happening all at the same time and has to be highly coordinated. Everybody's got to be on pace, on task, and in time and in tune. Think of it like a great orchestra, right? There's a lot of times and a lot of instruments are doing a lot of different things. They're not all doing the same thing, but everybody has to be doing exactly the right thing at the right time. Otherwise, it sounds like a bunch of noise. Same thing is true here. In order to get that kind of integration, you have to have a very sound and very potent regulation system. Who's the regulator of and the integrator of the orchestra? All you musicologists, of which there's none in the room. It's the conductor, right? The conductor is the one that keeps everybody like making sure that they are on the right page, that they are resting when they need to rest, and that they're chiming in when they need to chime in, and that they are as loud as he wants them to be, right? So the conductor has a lot of power and influence over how integrated that orchestra is. By the way, the composer has a lot to say about it too, doesn't he? Or she, because there are female composers. And so the way they write the music is instructions. It's integrated instructions telling each musician exactly what they need to do, right? And so the reason why we talk about all this stuff, why we talk about these different types of endocrine reflexes, why we talk about negative feedback and these regulatory controls, is because when you take a look at physiology, the key to understanding physiology is understanding control and regulation. That's the key. And there's a lot of it. Okay. And that's kind of, so we want to understand and lay our groundwork for our control and regulation in order to understand how all of this works and fits together. Once you understand the control and regulation of a system, the anatomy and all the rest of that stuff becomes easy because it makes sense. It's like, oh, wait a minute, if I need you to do this, of course you're going to look like this. You have to look like that, right? Okay, so let's go ahead and take a look at shall we? Because trust me, we're going to be talking about control and regulation all semester, so you're going to get it. So let's start off with the grand poobah. At the top of the food chain is the hypothalamus. And remember, in the hypothalamus is something like 14 different nuclei. And we're only really interested in a few of them. Some of these nuclei will be in charge of producing oxytocin and ADH. Those are the two hormones that are actually produced in the hypothalamus by these neurosecretory cells. So these guys right here are neurosecretory cells, whose cell bodies are in the hypothalamus making ADH or oxytocin. And then what they'll do is, just like a neuron traffics a neurotransmitter through the axon on those little neurofibrils, just basically walking it along and sort of stores those vesicles in the axon terminus. The same thing's happening here. The only difference is, instead of a neurotransmitter, it's ADH. And you're bundling that up into a vesicle in the cell body, and you're walking it along this axon fiber, where it gets stored in the pituitary, in the posterior lobe of the pituitary. Okay. So this is kind of like a two-part cell. We see this a lot in physiology. Remember in A&P1, we learned that there's 4 different types of tissues, right? There's epithelial tissue, connective tissue, muscle, and neural tissue. And the functions of them were discrete, right? So that is to say that epithelial tissue did epithelial stuff and neural tissue did neural stuff, but the two didn't mix. Well, here for the first time, and for the first time in several times that we're going to see throughout the semester, we have a dual role cell. We have a cell that's not just neural, it is a neuron, but it's also secretory. Secretory is the role of epithelial tissue. So this is kind of one of those switch hitters. It's kind of secretory like an epithelial cell, but it's still a neuron. Okay. So what it's doing then is making these hormones. These axons will go through this stalk-like structure of the pituitary called the infundibulum. And then it will move into the posterior pituitary gland where those vesicles full of ADH or oxytocin, depending on which neuron it was, will hang out. And they'll wait for the correct signal for their release. Okay, so they're just kind of hanging out there. And that's kind of what's happening. There are other types of nuclei as well in the hypothalamus as well. There are some sympathetic nuclei who have neurons that will actually communicate directly to the adrenal glands. So notice here is a neuron with its cell bodies in this sympathetic nucleus in the hypothalamus with a direct synapse tethered to the medulla of the adrenal glands. Why is this important? Because the medulla of the adrenal glands secretes your adrenalins, epinephrine and norepinephrine. So then, and some of you guys may have remembered this from A&P1. You may not have, that's okay. Because we're going to talk about it a lot here relative to the way it's actually at work in our systems. The sympathetic nervous system has a nickname. What is it? If I trigger your sympathetic nervous system, what are you ready to do? 2 things, fight or flight, right? It's your stress response. So when you get stressed out, when I let the lion in the room, or worse, when I let the exam in the room, right? That immediately kicks in a stress response and you have an entire physiological response to that. A lot of that response is adrenaline production, right? Because what happens if I bring the lion into the room? You're going to get an adrenaline rush, right? Which is the reason why people like that whole running with the bulls thing, right? You're going to get an adrenaline rush and your body is going to prepare for one thing: run like the wind, right? Because you want to make sure you live. Well, that's where your adrenaline response is. So what happens is when you lock eyes onto the lion, neurologically, you're communicating to the sympathetic nucleus to fire, and it's immediately going to tell the adrenal medulla to start producing adrenaline quickly because you need to start changing your body's physiology to run like the wind. Okay, so it's a direct connection. This is a direct neuroendocrine connection with this one. So in your posterior pituitary, then once you actually move this, and there's a lot of other nuclei in there, and we're not going to get into those, but these are important ones because it kind of shows us how our endocrine system is tethered to the hypothalamus. Now, once we have our ADH and our oxytocin stored in the posterior pituitary, now we can focus our attention on the anterior pituitary or the adenohypophysis. By the way, the hypophysis is the technical term for the pituitary. You can use either term. If you want to use anterior pituitary or adenohypophysis, that's fine. They're synonymous terms. You will see them equally, so you should be familiar with both, because in some sources you'll see the hypophysis term, and in some you'll see the pituitary term. So this is your adenohypophysis. And the posterior would be your neural hypothesis. And so those are your two. By the way, if you're recalling the structure that your pituitary is in, if you remember from AMP1 when you guys looked at the skull bones, you remember the sphenoid bone, right? That was the one that looked like a bat. That's like right about here. So if you take your fingers and you point like this, the connection point is about where your pituitary would be, resting comfortably in that little divot in the sphenoid bone called the hypophyseal fossa. Right? And so that's the reason why they call it the hypophyseal fossa, because the hypothesis is the technical term for the pituitary. The pituitary is rest right in that little area inside your sphenoid. Now, what is the anterior or the adenohypophysis doing? So in this cell, in this side of the pituitary, you've got a bunch of cells who are secreting and releasing. So there's 7 different hormones that they're secreting, and then they're going to be releasing those hormones out to the body, to their target glands, and so forth. So altogether then, you have 7 hormones on the adeno side being made and released. You have 2 that are made in the in the hypothalamus, but stored in the posterior pituitary for a total of 9. Okay, overall. Only 7 of which are made by the pituitary. So these 9, and we'll take a look at each one of the 9 in just a second here. But these will control. These are like the command and control of the endocrine system throughout the body. Okay, so this is the initial orders. From here, it's kind of like you tell your people to tell their people to tell their people to do this, right? So you kind of have this cascade of command, just like you have in the military. And that command and control is these reflexes. Now, what happens when both neural and endocrine combine? Well, we call that one a neuroendocrine complex. So this is where you have both neural components and endocrine components. A perfect example of that is what we just saw with your sympathetic. This is a neuroendocrine pathway. It's got a neural connection in the hypothalamus, right? Because those where the neurons are. And then it basically is a synaptic type of communication with the endocrine component, which is the adrenal glands. Okay, so that would be a neuroendocrine. You're combining the activities of both nervous system and endocrine system in order to regulate. So ultimately then, when you take a look at hormone release, as you're releasing, if you're the pituitary and you're releasing hormones out to the rest of the body, a lot of times we looked at simple release versus complex release, and so you can get a lot of complexity and it's kind of like shades of function with that. But a lot of times you can also get a lot of complex commands with not just what you do, right? Not just the amount. That's one way to control your regulation. So like, for instance, a little bit of hormone has one effect, a lot of hormone has a different effect, right? Testosterone is that way. A little bit of testosterone has one effect, a lot of testosterone has a very different effect. Okay, so that is a controller or a complexity associated with the amount. Okay, the other one is also the pattern. So for instance, you don't have to secrete your hormone all at once. You can sort of do these patterns or these temporal patterns. Like maybe you'll do an injection here, and it just like a series of them of secretions, and then or versus maybe like a long-term consistent secretion. So you can get 2 different effects based on the pattern of how you release it and how often you release it. Right? Think of like a dam. I mean, one that we're in the pathway of, right? The Cherry Creek Dam, which is a flood control dam. You would imagine that the Cherry Creek Dam is releasing water all the time, but the pattern that it uses to release that water can have very drastic effects, can't it? If it releases it in a little trickle, we almost recognize no water whatsoever, right? If it releases a lot of water, then we're going to notice a big difference. If the Cherry Creek Dam just basically breaks down the dam and just lets the entire Cherry Creek Reservoir do what it wants to do and just come down the Cherry Creek bed, we're going to notice it, right? Most of Southeast Denver will be underwater. So you can see very different effects based on how you do that time release. And you can get those same types of complexities in your command structure with your reflow. Now, why do you say that? I say that because in order to integrate all the stuff that's going on in your body, I mean, all of the amazing things that are happening in your body all at the same time, you've got to have a lot of complexity involved in there. Okay? There's got to be a lot going on in there because everybody has to know what they're doing and when they're doing it and for how long. So that's the reason why you have to have so much control and regulation. Okay, let's take a look at the pituitary gland proper. So this is our hypothesis. It's going to be hanging out inside the cella turcica. So basically, if you take a look at the cella turcica, remember this basically means the Turk saddle. So if you guys remember your sphenoid bone, you kind of had that little flat anvil shaped kind of section of the bone, and you kind of had the little hypophyseal fossa like that. So that is the cella turcica, that little anvil area. And the pituitary actually rests right inside that little cella turcica. There's your little infundibulum. And then it's covered with what's called the cellar diaphragm. And this kind of keeps your pituitary in place, keeps it from sort of jostling around, and kind of secures it a little bit. It's also outside your cranial cavity, right? Because on the upper portion up here is going to be where the hypothalamus is and the rest of your brain. So this kind of seals it off from that area. And that's the reason why, like, for instance, when we saw the sheep brain last time, we didn't see the pituitary, did we? Because generally speaking, when you go and butcher a sheep, you're probably just taking the brain out of the skull case. But if you don't take the sphenoid with it, you're likely just going to rip that pituitary off. And so you have a little sheep brain that doesn't have its pituitary anymore because the pituitary is still in that hypophyseal fossa, whatever they did with that sphenoid bone, right? That's the reason why you didn't ever see that pituitary in the sheep brains, is because of that. So it's basically connected by your infundibulum, and of course we already mentioned that it releases 9 hormones, but makes 7 of them. The other 2 are made by the hypothalamus, right? So that's oxytocin and ADH. So basically, when you're looking at the release, especially of a lot of these, most of these are coming off the pituitary are peptide type hormones. So these are the peptide types, which means that generally speaking, when they're released in the bloodstream, they have to go and communicate with cells. And generally speaking, when you're looking at communicating with cells, what you usually have is a receptor system. So where your hormone will come in and it'll bind to your receptor, and then that will trigger some sort of an intracellular mechanism that will communicate with the cell to do whatever it needs to do. Generally speaking, when you look at that process, by the way, this whole process here, this whole idea of a cell internalizing this hormonal signal on the outside is something that's called cell communication. No. So there's different ways to do this. There's multiple patterns. One is like what's called using a second messenger system. So basically this message gets relayed to another, like a go-between. It's kind of like the Pony Express. You know, you sort of pass on the message to your messenger, your courier, and it's their job to take it and run. That's like a second messenger, right? Second messenger system. And there's other strategies to this, and you'll see this in your chapter. We don't have a lot of time to do that because this is this is like that cell transmission, that direct cell transmission idea. That is a really, really big topic. We can spend an entire chapter actually just talking about that alone. But there's different versions of this. One's called like what's called G protein coupled, where there's a specific type of a protein that is relaying this message. But at the end of the day, regardless of how you structure it, the basic thing is the same. You're relaying the message to the inside of the cell so that the cell can do some sort of a mechanism, right? So the signal transmission is a really, really big thing. And so I don't want to get into it. I just want to let you know that there's a thing here, right? There's an entire chapter that's there when you're talking about internalizing that signal and the ways that you do that. And the reason why it's important is because we actually develop drug therapeutics based on that signal transduction pattern, right? Whether you use cyclic AMP as a second messenger or if you use the G proteins, different types of strategies will allow us to be able to develop different types of drug therapeutics for a particular response. So that's the only reason why I mention it, is because there is a pretty large group of class of drugs out there that are looking at signal transduction types of strategy. So here's our pituitary. So here you can see your anterior lobe. No, not from start. So here you can see your anterior lobe on this side. That is your adenohypophysis. And of course your posterior, which is your neurohypophysis on this side. And in the middle is what's called your pars intermedia. So the adeno side, the anterior side, is broken up into a couple of things. So the stalk portion of the adeno that goes up and sort of forms part of the infundibulum is what's called the pars tuberalis. And that's kind of part of the stalk that kind of dangles down. The bulk of the secretory cells that you see in the adeno is pars distalis. And then in the middle is pars intermedia, which is, you can actually see the structural change. So when you look at the pituitary slides in lab, you can actually see the change in the cell structure and the cell texture from anterior to intermediate, the pars intermedia, to the posterior or the neurohypophysis, because structurally they're doing very different things. And of course, you can see here's your cellular diaphragm, which basically pinches these guys apart and closes it all off. So all your communication between hypothalamus and your pituitary is happening through the infundibulum. So everything's going through here. So this is what it looks like. This is actually what you're pretty accurate. So you can see the pars distalis over here. Notice now the classic look of a gland, right? This is to kind of go back to your epithelial, your AMP1. But notice what you have is clusters of cells that are cuboid in nature. Whenever you see cuboid cells, most of the time, you're looking at a gland, especially if you see a cluster of them. But you can see there's like a big cluster here of lots of little cuboid cells. The peak is the nucleus of those cuboid cells. And so you can see they're very cuboid, typical of what you would expect to see from a gland who is secreting something, right? So these are cuboid clusters. Then on the posterior lobe, you notice it's a very different look. You notice this is a much more fibrous look. Why? Because what you have in the neuro side is mostly axons from your neurosecretory cells. And axons have that white fibrous look, right? Remember, we learned this in AMP1 when we learned about the difference between gray matter and white matter. So it looks very distinct and looks exactly the way you expect it to look if you've got a bunch of axons in there. And then, of course, if you look at the pars intermedia, you can see it looks somewhat sort of intermediate between the two. Not quite as white and fibrous as the posterior, not quite as cuboidally cellular as the adeno or the anterior. So it's kind of like an intermediate look. And that's your pars intermedia. So on your adeno side, these guys are secreting 7 and releasing 7 of the pituitary hormones. The posterior is only working with ADH and oxytocin. And what does pars intermedia do? Pars intermedia actually secretes a hormone called MSH, which is melanin stimulating hormone, which is something that you create early on, and then it's not really active in adulthood. But this is something that is part of your melanin production system, MSH. And it's usually like when I say early on, I mean like fetal development, right? So it's early on. So let's take a look then at our adeno hypothesis. So in this particular case, when you have hormones, they have to turn on your endocrine glands. Seriously. All right. And so once you turn on these hormones, and they will start activating these regions. So obviously tuberalis is part of the infundibulum. Distalis is the main body. And the intermedia is basically the demilitarized zone, the DMZ of the pituitary, right? Separating both anterior and posterior. Now, one of the things that we see in the hypothesis, and we see this again, these are important systems, is what's called a portal system. So what a portal system is, and we see 2 of them. When we see portal systems, there's not a ton of them, but when we see them, they're very critical. So what a portal system is, and I'll define this here in just a second, but what I want to do is I want to define a little bit, first of all, about how circulation works in your body. So generally speaking, when you take a look at circulation, it starts from your heart. It gets pumped from your left side to somewhere in your body through arteries, and the arteries will break down into capillaries. And then the capillaries will basically turn into the venous side, and then it'll go back to the heart for redistribution. So there's a capillary bed. Now, if I wanted to move blood from that capillary bed to this capillary bed, what I would have to do is I would have to go all the way back to the heart, get pumped out the left side, I have to go down arteries, and I have to go back into this capillary. So I had to go all the way back through the heart. That's how normal circulation works. A portal system is when 2 capillary beds are connected. It's like a short circuit by a portal vein. So this is when two capillary beds are connected to each other directly through a portal vessel. What this does, it allows the blood from one capillary bed to move immediately to the other capillary bed without having to go all the way back to the venous system, all the way back to the heart, and then get pumped all the way back to the arterial system to the next capillary bed, right? This is particularly useful when you have an area where you're kind of like working on something and you're not ready for general broadcast to the body. Because when you go through the heart, you're broadcasting to the whole body, right? So think of this as, and this is like a lot of messaging. I mean, you've got a message that you're working on, right? But you're not ready to release it to the general public because you're still working on it. A lot of issues of policy at the higher levels of government are this way, right? Like the president's office doesn't just release everything that they're talking about and working on. Why? Because there's things they're trying to hammer out, and there's things that are going to change, and they're trying to figure some things out. So what they have is direct communication between the parties that are involved, right? Between the president, maybe the leader of the Senate, and the leader of the House, and they're involved with each other, but they're doing meetings and they're trying to work out things. And then once they figure it all out, then they release it to the public so that it makes sense to them. But while they're working on it, it's just them working on it. So they have direct communication between each other without broadcasting it to the rest of the body. And that's basically what a portal system does. It allows, in this particular case, for the hypothalamus and the pituitary to discuss matters of business between each other before releasing it all out to the body. And that's what that's the portal system does. So there is a couple of vascular beds that are associated with the hypothalamus, and then there are also a couple that are associated with the pituitary as well. And so generally speaking, what will happen is through this system, the hypothalamus will communicate with the pituitary using regulatory factors. And it'll move this into interstitial fluids, which is basically the fluid around the cells in the tissues. And then it'll put these out in the interstitial tissues. It'll be taken up by fenestrated capillaries, and then it'll be moved to the pituitary. And that's how the hypothalamus is giving instructions to the pituitary to do its thing. And so this is just between them, right? This is them coordinating between each other. This is like the king talking to the prime minister to coordinate matters of state. It's between them, right? And so this is all happening through the median eminence, which is, I'll show you, we had it over here. So this is the median eminence here. So that little bottom portion of the hypothalamus is the median eminence. And that's where most of these communications are going as the hypothalamus is giving instructions to the pituitary. So then there is a lower region or a lower network in the pituitary. So your two capillary networks are connected with this portal system to create your, or with the portal vessel to create your portal system. And this basically allows you to regulate everything. So here's what it looks like. This is your hypophyseal portal system. So right here is one capillary bed. And then here is the next major capillary bed down here. And so here you can see there's a lot going on here, right? So here you can see that you've got neurosecretory cells in the paraventricular nucleus, neurosecretory cells in the supraoptic nucleus, which are sending their material through the infundibulum into the posterior pituitary. And notice their connections, their synaptic connections are to blood vessels, which are associated with a capillary bed. So then what's going to happen is they are going to release oxytocin when they need to, and they're going to release it into the bloodstream, or ADH when they need to. And they're going to release it into the bloodstream, immediately into the bloodstream. But in this particular case, what's going to happen is notice these guys here are synapsed to the capillary bed. So you can see they're communicating directly to the capillary bed. So this is where these communications are coming. And then it's being connected through this guy right here in particular, which is your portal vessel. And so you're connecting this capillary bed with this capillary bed directly with that portal vessel. So that the hormones that the hypothalamus are making are going directly to the adenohypophysis to give them instruction, like it's not for everybody else, right? Because everybody else isn't listening for the hypothalamus. Everybody else is listening for their immediate commanding officer, right? That's what they train you in the military to do. You listen to your CO, and they're supposed to listen to their CO. So if everything goes well, the instructions that the top guy gave are exactly the same ones you're going to hear when you have boots on the ground. Okay. And that's basically what a portal vessel does. It's connecting these two capillary beds together so that you can get direct communication between the hypothalamus as it's dumping its hormones into this capillary bed, and it goes directly into this capillary bed to instruct these cells. That's how the adenohypophysis cells know what to do. They know which of the 7 hormones they need to make. Why? Because they're getting those instructions from the hypothalamus through this portal system. Okay. So what about these hormones that the hypothalamus is sending? What exactly are they? How do they work? Well, there's 2 of them. There's releasing hormones and there's inhibiting hormones. So releasing hormones basically are what they say they are, right? These, when, for instance, the adenohypophysis gets a releasing hormone, it's going to be told to make and secrete the hormone that it's designed to make and secrete. Okay, it's that simple. On the flip side, the inhibiting hormone is just the opposite. It basically stops the synthesis and release of these hormones. So you basically have an on-off switch. This is the on, this is the off. Now, how do you control these signals? You control it again with regulation, right? So one of the regulation or regulatory types of patterns that we see is what's called a feedback loop, and in particular, negative feedback. So let's take a look at what feedback looks like. So feedback itself is basically a situation. I want to kind of describe feedback a little bit. I want to do it on this slide because feedback is a regulatory strategy, but it's something that is a little easier understood. Like if I give you like a biochemical pathway sort of a thing, because a biochemical pathway is very stepwise. It's very discrete. It's very easy, very simple to understand. Okay, and it'll also do the trick of explaining how the 2 different types of feedback work. There's both negative and positive. I want to start off with negative feedback. In a negative feedback situation, this is where a product inhibits its own production. So if I use the example of an enzyme, like an enzymatic pathway, say I start off with substrate A, which gets converted to B, which is converted to C, which gets converted to D. And D is the final thing, right? So it's a multi-step pathway. And enzyme A does this reaction, enzyme B does this one, and enzyme C does this one. In negative feedback, what happens is that as D accumulates in high concentrations, what happens is it will basically go back and inhibit its own production. It's kind of like a fail-safe. And that little flat T is the inhibitory or the inhibition signal. It's simple. Okay. So it basically turns itself off. Now, in positive feedback, it's just the opposite. This is basically where a little product stimulates its own production. So let's use the same situation. A to B to C to D. In this case, imagine this is a situation where like you're getting a little bit of D, just a little bit, like a little trickle of D in there. And then once you start to get that little bit of D, it feeds back and reinforces its production so it makes more. That's a positive feedback. So why are these important? Well, let's take a look at what it looks like. And I'm going to start with positive feedback first. Now, when I have a positive feedback loop, the reason why positive feedback loops are important is because imagine that I had some sort of a protein and I needed a certain amount of it in order to survive, right? Like something happened and I need to make a lot of it in order to save myself. But normally speaking, it's just, it gets made very slowly. I'm making a little bit of it. I'm getting there. But let's say you have to get up to here in order to survive. Guess what? You're not going to live, are you? Because you're never going to get there, right? By the time you get up to that level, the lion will have long eaten you and picked its teeth and is ready for its next meal, right? So in a positive feedback situation, what happens is this little trickle down here at the bottom stimulates, reinforces its own, sorry about that, make you guys see sick here, reinforces its own production. So once that reinforcement comes in, it doubles down on production, upregulates it. So instead of being flatline like this, as soon as that little bit comes in, you start to see it go like this. It goes up very quickly. So within a very short period of time, now all of a sudden, you've made your limit. Guess what? You get to survive. But guess what? You don't want to stay there, do you? You got all this product all over the place. The lion's gone now. We're good. Guess what you have to do now? Bring yourself back down, right? So what do you have to do? Now what you can do is you can link a negative feedback loop to this on the back end and say, you know what? Once you get up here, we're going to kick in a negative feedback loop, and that's going to shut you down. So now it's going to bring it all the way down, and it's going to look like this. So on one side, you've got the positive response, rapid rise in production to get you to where you need very quickly. And then on the back side, you've got your rapid reset. Bring it all back down because you don't need all this extra protein laying all over the place. So a good way to do that is to use your negative feedback loop to inhibit your production, and then you can just clean up everything else that you've got. Okay. That look familiar? The pattern looks very similar to what we were looking at in AMP1, right? We had some sort of a rapid rise in response and then a recovery. A rapid rise in response and then a recovery. Similar situations happening here. So positives and negatives are necessary. So it's kind of what it looks like when we take a look at our feedbacks and also our releasing and inhibitory protein. So for instance, if you just take a look at thyroid, right? So basically coming off of the hypothalamus, we've got thyroid releasing hormone, which is being made and produced. It's going through the portal system, and it's going to be going to the anterior lobes. That's a releasing hormone telling those anterior lobe cells to produce and release the thyroid hormone that they're responsible for. And what is that? It's not thyroid hormone, it's thyroid stimulating hormone, TSH. So that's what the releasing hormone tells them to do. So make TSH and release it. And what do? They make TSH and they release it to the bloodstream. It's an endocrine situation, right? And so that hormone goes to the endocrine gland, which in this case is the thyroid. And the thyroid has the necessary receptors to receive that hormone. A lot of times students will ask the question, it's like, well, wait a minute, if you've got TSH going throughout the rest of your body, then what are the other cells doing with TSH, right? I mean, if everybody's seeing this message, then what is the other cells doing with TSH? The answer is they don't see it. Because the only way that a cell can see a message or a signal is if it's got a receptor for that message or for that signal. If it doesn't have a receptor, it is blind to the signal. So you can bathe that cell in as much signal or hormone as you want. And if that cell doesn't have the receptors for it, it's not going to do a thing. So what that means is the only cells in your body that have the receptor for TSH are found in your thyroid. So they are the only ones who actually respond to the TSH signal. That turns these cells on and causes them to make the thyroid hormones, T3 and T4, which then are released by the thyroid gland to the target cells to tell them to do their metabolic activities. Okay. Now, where does it? That's a normal forward flow. That's a normal sort of a hormonal flow. That's a pretty typical one, where you have multiple stops, right? So you basically go to a target gland, and the gland will then send it to the target cell. So it's kind of like a little bit of an intermediary there, and that's a common thing. Not all hormonal pathways look like this. Some of them do. This is what's called a tropic pathway, because you make an intermediate hormone. Okay, so what's coming off of the pituitary is not going to the target. There's some other work to do. Okay, that's what's called a tropic situation. Where does the feedback come into play? Well, T3 and T4, for instance, will actually negatively feed back. So if you have a lot of T3 and T4, it'll actually shut down the secretion and negatively impact the anterior lobe secretion of TSH. It'll also negatively impact the hypothalamus and shut down the production of releasing hormone, right, which shuts down the entire pathway. So here you can see that the accumulation of your terminal hormone, T3 and T4, the thyroid hormones, will actually shut down its production. Okay. So what do some of the other ones look like? So this is what's called a non-tropic version. So you notice there's not an intermediate gland in there. It's just one target. It's very simple, straightforward. So prolactin, which is for lactation of the mammary glands. When you take a look at prolactin at the hypothalamus level, you can see you've got both inhibitory and releasing factor. So if you make the releasing factor, then you will stimulate the anterior lobe to make prolactin, which will then stimulate directly the mammary gland tissue to actually produce lactation milk. However, the inhibitory will inhibit the anterior lobe and will shut down prolactin production. Now, how does this work in terms of stimulation? Well, when you have a lot of prolactin in your system, this will actually shut everything down. So for instance, a lot of prolactin, this is a negative feedback loop, right, will basically feedback negatively on the releasing factor, which undercuts the activation signal. So you're not making any more prolactin. It will also stimulate the inhibitory activity, which will put the brakes and make the brakes much stronger, thereby cutting down on the prolactin and ultimately the secretion of milk. Okay. So you can see a couple of things here, and this is a common thing that you see in regulation. One way to stop a response is either to apply the brakes or to disable the accelerator. That make sense? One way to slow down the car is either passively or actively. This one here would be passive. This is like you taking your foot off of the accelerator, right? You're basically inhibiting the thing that's making you go forward, which slows you down. Or what you can do is actively slow it down. The stopper, by stimulating the stopper, which is your brake pedal, you are basically actively inhibiting or slowing down the car. Either one will slow the car down. So you can either inhibit the activator or activate the inhibitor. Wait a minute, did that make any sense? That's not like I said the same thing twice. You can either inhibit the activator, right? Or activate the inhibitor. Inhibiting the activator is you taking your foot off the gas to slow the car down. Activating the inhibitor is you putting your foot on the brake pedal to grab the car, the calipers, and slow it down actively, physically. That makes sense? You see that actually a lot in regulation, a combination of the two. Okay. So that's one where you don't have an intermediate, right? So basically what's coming off of the pituitary is going directly to the target cells in the mammary glands. That's a non-tropic type of a strategy. And so let me just write that in there. So tropic. So these are tropic pattern. By which I mean uses an intermediate gland. As non-tropic. In this case, you don't use an intermediate gland. Your pituitary hormone goes directly to the target. And then there's a combination of the two, which is the growth hormones, which gets really interesting because you have both pathways, tropic and non-tropic. So let's take a look at it here. So first of all, you've got your inhibitory for your growth hormone and your releasing, right? So if you release, then that's going to cause your anterior lobe to make growth hormone and release it. And that growth hormone will be made. And then at this particular point, it can go into either a tropic pathway or a non-tropic pathway. So if the growth hormone goes to the liver, which it can do, and it makes things like somatomedins and so forth, which can then go build, like, for instance, skeletal muscle, bones, that sort of stuff, then this would be sort of like a tropic pathway. Why? Because the liver is acting like an intermediate gland before it gets released to the body. However, growth hormone can go directly to tissue and communicate and deal with them. This would be like the non-tropic. So like epidermal growth factor, for instance, which is the one that keeps your stratum basale dividing so that you always have a new skin layer replacing your skin, would be a version of this non-tropic pathway where your growth hormone is coming off and it's going immediately to work at the target tissue. Okay, so you have both with growth. Now, if you have an accumulation of these, especially like the somatomedins, then an accumulation will basically shut down your releasing hormone, right? So that's inhibition of your activator. That's taking your foot off the gas. And then it'll also actively stimulate the inhibitor, which is like you pushing the brake pedal. That makes sense? So growth hormones, you can see it's a combination of the two. So these are very, very common regulatory mechanisms in terms of controlling how these things work. Okay, let's take a look at this one. This is a really, really nice, I love this image. There's a lot going on here, obviously. this is, I like this image because a lot of times students will be like, wait a minute, there's like so much going on here. How do I study for this chapter? Well, one thing I always tell students to do is grab a hold of images that have a lot of density information on them. Because it's easier for your brain to, you kind of fool your brain into thinking you're only studying 1 or 2 images as opposed to the entire chapter. So if you can remember this with a lot of information on it, then you've got a lot of endocrine in your head. Okay. The reason why I like this is because it's like a visual table of contents for pretty much the entire endocrine system. This is a huge chunk of the chapter. Okay. So it shows you your pituitary and what all the pituitary is actually making. And so we're not going to talk about all these in super in-depth, but we're going to cover them lightly. We'll talk a few of them a little bit more in depth. So first of all, one of the things coming off of the adeno hypothesis, that's all the purple ones here, is adrenocorticotropic hormone, that's ACTH. And so this will actually go and stimulate a lot of the areas of the adrenal gland. And we'll take a look at its role in the adrenal gland. We'll take a look at the adrenal gland specifically and crack that one open a little bit here in just a second. It makes TSH. We learned about this one in the previous slide, and that goes to the thyroid gland. The thyroid gland will basically make T3 and T4. Whereas when ACTH goes to the adrenal cortex, it'll make either adrenalins, or it'll make a lot of these corticosteroids and things like that. Okay. So you have 2 main connections to the adrenal glands, ACTH to the pituitary, and then of course you've got that direct sympathetic nucleus directly linked to the medulla to make adrenaline, right? So your adrenal glands actually have a lot of important connections when it comes to that. The liver is going to be taking growth hormone from the pituitary and turning that into somatomedins, which will then grow your bones and your muscle, which is why we call them growth hormones, right? Makes them larger. Coming directly off of that and going directly to the tissue is prolactin, which is also coming off of the pituitary. And then we have FSH and LH, so luteinizing hormone and follicle stimulating hormone, also coming off the adrenal hypothesis. Now, notice you have a couple of things going on here, right? So you have, notice FSH is both working on the ovaries and also the testes, right? LH is working on the testes and also the ovaries as well, right? So we oftentimes think of these as the female hormones, but they're actually more like reproductive hormones. It's true for both males and females. So what's FSH and LH doing? For the most part, is a lot of what it's doing is organizing things like spermatogenesis and oogenesis. So the whole meiosis process that's going on in oogenesis, spermatogenesis, a lot of follicle stimulating hormone and luteinizing hormone are associated with that. And so that's part of that whole kind of processing gametes. That's kind of what FSH and LH are doing. And we both need to process gametes, right? That's not something that's just a female thing. It's not just a male thing. It's both, both individuals. Now, in response to FSH and LH, we do different things, right? So for instance, in females, you're going to be making estrogen, progesterone, and inhibin, whereas in males, it's inhibin and testosterone. So slightly different spectrum of secondary hormones that are coming off of our ovaries and testes. But the idea is governing and regulating the production of those secondary hormones is what FSH and LH are doing, and that's something that we both need. So this is for you table people, and this is purely organizational. Some of you like images, and that's fine. Some of you like tables, and that's also fine. This is basically just kind of putting everything in one place for you. That's all this is. So it's just a study aid. And I'm not going to really go through stuff like this. That's just there for your use and for your convenience. Okay, so that's the adeno. Oh, by the way, we missed one, didn't we? MSH, which is the melanocyte stimulating hormone. That's only made by the pars intermedia. And so that's the production of melanocytes. So that's early on. So that's just like embryonic development of those melanocytes. Okay, now let's take a look at the posterior, right? So this is our neurohypophysis. And so this is basically where we have our axons that are holding that ADH and the oxytocin. And this ADH and oxytocin are made in two different areas of the hypothalamus, the paraventricular nucleus and the supraoptic. So the supraoptic is the one that's responsible for making oxytocin. Excuse me, got that backwards. Always get that backwards, actually. The paraventricular is responsible for making oxytocin, and the supraoptic is responsible for making ADH. Okay. And then they'll make these, and then they'll store them, just like we saw before, right? The hypophyseal portal system in those little terminal axonal areas tethered to the blood vessels, ready for release whenever they should need that release. So the ADH, when it's needed, is going to go to the kidneys, and of course the kidneys, we know, will make calcitriol. And oxytocin is going to be going in two directions, or one direction, because you can't be both male and female at the same time. So oxytocin will either go to the males. In males, basically, this is for the smooth muscle contraction of the ductus deferens for the movement of semen through the duct system in males. So that's what oxytocin does there. And then in females, it's basically for uterine muscle contraction of labor pain. Right, so those labor contractions are stimulated in a feedback loop, a positive feedback loop by oxytocin. And so that's probably the most famous role for oxytocin is during labor. And it's probably one of the most famous positive feedback loops, actually. And that's the reason why, by the way, in labor, it has that sort of, that kind of progressive personality to it. Like when the first contraction starts and that oxytocin hits, then those contractions start to get closer and closer together, and they kind of start, it kind of like starts to move quicker and quicker, and it kind of starts off slow, and then it kind of starts to move. What you're feeling is that rapid rise of the positive feedback loop that's being triggered by oxytocin release. And that's one of the most famous ones. And of course, then once the baby is born, then all of that oxytocin production kind of goes into a negative feedback loop, so it feeds back negatively, and then it stops the contractions because they're no longer needed. And the same thing is true for the musculature in the male reproductive system as well. It's a similar type of a scenario. But that's where oxytocin and ADH come into play. We'll have a lot more to talk about ADH. But in addition to all of this, you also have some connections to osmoregulators and sensory neurons that are in the hypothalamus, right? So the reason why I want to mention that osmoreceptors is basically you've got lots of connections, not just outputs, but lots of inputs. Because remember, the standard protocol is to take in sensory information, to process it, and to make some sort of a decision in terms of an output, right? Normally in AMP1, what we're used to is something that's physically sensory, right? Like I touch the table and I feel that it's smooth. But on a physiological level, most of those inputs you're not even aware of, right? So those inputs, those sensations that are coming in are more things like, is my blood pressure getting high? Well, you don't know that necessarily. Could be high, could be not, right? But you've got sensors whose job it is to sense that, whether or not that blood pressure is getting high, or whether or not the blood pressure is getting low, or whether your calcium is getting high, or whether your calcium is getting low, or whether your blood glucose is getting high, or whether your blood glucose is getting low, right? Most of these things you're not even aware of, but you've got sensors in you that are communicating constantly, collecting information on all of your internal measurable metabolites that are generally under homeostatic control, and they're feeding that back to these control centers. And these control centers are autonomically making some sort of a decision about what to do in response to that information. So that's why I say the difference between endocrine and neuron is you're aware of your reflexes usually in your neural reflexes. Usually you're not aware of your endocrine reflexes. If everything goes well, you have no clue what's going on with your endocrine reflexes until something goes wrong. Then you realize, wait a minute, something's not working. So this is the finish up to the table for the posterior lobe. Okay, any questions on pituitary and pituitariness before we dig into thyroid? Feeling good about it? Okay. Let's dig into the thyroid and just at least kind of get it introed, and then we'll kind of dig into some of the heavy lifting. So the thyroid gland basically is going to be sitting on top of your thyroid cartilage. Where's that? It's your gully whopper, right? So the big thing here, that's what my dad used to call it. He was not an anatomy guy, right? So that's this guy right here. So it's resting right there. And basically it's like a big butterfly with two lobes as the wings and the isthmus as the body. And inside the tissue, the actual tissue is composed of what's called thyroid follicles. These are the functional units of the thyroid itself, which basically look like these little cuboid wheels. These spheres of cuboid cells around a cavity with this sort of viscous colloid in it. And then wrapped around that, you've got capillaries and stuff like that, right? Because they're making T3 and T4, the thyroid hormones, and they're dumping that into the bloodstream for distribution to the body. So you would imagine then that you've got lots of capillaries in there that are designed to accept these secretory products and basically move them on out to the rest of the body for broadcast. And they're also going to be picking up waste. They're also going to be delivering nutrients and other types of hormones. For instance, TSH is going to be coming through these capillaries. So remember, capillaries aren't just to offload your material onto, it's also to deliver stuff to you. Right? So it's like DoorDash. It's bringing you food. Only DoorDash doesn't pick up your trash. But it's bringing you food and nutrients and oxygen and everything you need as a cell group. But at the same time, it's also taking your trash and it's taking the hormones that you're offloading into the bloodstream for distribution to the rest of the body. So it's a combination DoorDash, trash collection, mailman. That's basically what your capillaries are. If you combine those three services into one entity, you would have essentially what a capillary is. So there are three things that are going to be made in the thyroid gland. We're going to talk about those guys, but because we are at about our witching hour, I'm going to stop there. I'm going to pick up with that, and then we'll take a look at the structure and especially the histology of the thyroid. So this should help a lot with a lot of your histology slides that we're working on in lab as well. But we'll take a look at the thyroid gland and what in more depth in terms of how it secretes and releases its hormones. Okay, so let's go ahead and stop there. And then take a little bit of a break.